Good day, ma'am. Today I will be discussing a critical part of cellular respiration, which is the electron transport chain, or ETC. This is the fourth stage in the process that allows our cells to produce energy in the form of ATP. So what is electron transport chain? Well, it is a series of biochemical reactions in which electrons and hydrogen ions from NADH and FADH are passed to intermediate carriers and then ultimately react with molecular oxygen to produce water. The NADH and FADH produced in the citric acid cycle pass through the electron transport chain. NADH and FADH are oxidized in this process. The electrons that pass through the various steps of the electron transport chain lose some energy with each transfer along the chain. Some of its lost energy is used to make ATP from ADP. The enzymes and electron carriers needed for the ETC are located along the inner mitochondrial membrane. Within this membrane are 4 distinct protein complexes, each containing of the molecules needed for the ETC process to occur. So, the electron transport chain is a series of protein complexes and molecules embedded in the inner mitochondrial membrane. Its primary role is to transfer electrons from energy carriers such as the NADH and FADH to oxygen, which is the final electron acceptor. This process generates a proton gradient across the membrane, driving the production of ATP by ATP synthase. Now we move on to the 4 protein complexes. Complex 1, NADH coenzyme Q reductase. Complex 2, succinate coenzyme Q reductase. Complex 3, coenzyme Q cytochrome C reductase, and complex 4, cytochrome C oxidase. These 4 complexes are tightly bound to the inner mitochondrial membrane. 2 electron carriers, coenzyme Q and cytochrome C, which are not tightly associated with any of the 4 complexes, serve as mobile electron carriers that shuttle electrons between the various complexes. What is a mobile electron carrier? Well, it is a molecule that transfers electrons between different protein complexes within the electron transport chain, such as your coenzyme Q and cytochrome C. Now let's dive into complex 1, also known as NADH dehydrogenase. This is the largest protein complex in the ETC and is the starting point for electron transfer. Its main function is to oxidize NADH, which carries high energy electrons from earlier stages of respiration, such as glycolysis and the citric acid cycle. This contains more than 40 subunits, including the B vitamin containing flavin mononucleotide and several iron-sulfur proteins. The actual electron transfer process is not, however, a single-step directed transfer of electrons from NADH to CoQ. Several intermediate carriers are involved. When NADH reaches complex 1, it donates 2 high-energy electrons. These electrons are passed through a series of redox reactions involving flavin mononucleotide and a chain of iron-sulfur clusters within complex 1. These clusters act like tiny stations, transferring the electrons step by step through the protein. NADH supplies both electrons and one of the hydrogen ions that are transferred. The other hydrogen ion comes from the matrix solution. The actual changes that occur within the structure of flavin mononucleotide as it accepts the 2 electrons and 2 hydrogen ions are shown below. The next step involves the transfer of electrons from the reduced flavin mononucleotide through a series of iron-sulfur proteins. So as the electrons flow through complex 1, the energy released is used to pump electrons, or hydrogen ions, from the mitochondrial matrix into the intermembrane space. This creates a proton gradient, which is like a reservoir of potential energy that will later drive ATP synthesis. The two atoms of flavine mononucleotide are released to solution as two hydrogen ions. The two iron-sulfur protein molecules are needed to accommodate the two electrons released by the reduced flavine mononucleotide because an iron reduction involves only one electron. In the final complex 1 reaction, ferrous sulfur protein is converted into ferric sulfur protein as each of two ferrous sulfur protein units passes an electron to CoQ, changing it from its oxidized form to its reduced form. So, by the end of this step, NADH is oxidized to NAD, ready to participate in other metabolic processes. Meanwhile, the electrons are passed to ubiquinone, also known as coenzyme Q, which will carry them to the next complex in the chain. Coenzyme Q, in both its oxidized and reduced form, is lipid soluble and can move laterally within the mitochondrial membrane. Its function is to shuttle its newly acquired electrons to complex 3, where it becomes the initial substrate for reactions at this complex. The Q in the designation coenzyme Q comes from the name quinone. Structurally, coenzyme Q is quinone derivative. In its most common form, coenzyme Q has a long carbon chain containing 10 isoprene units attached to its quinone unit. The actual changes that occur within the structure of CoQ as it accepts the two electrons and the two hydrogen ions involves the quinone part of its structure, as is below. The two hydrogen ions that CoQ picks up in forming CoQH2 comes from the solution. So this step, which is complex 1, is vital because it initiates the electron transport chain, setting the stage for ATP production. Any dysfunction in the complex 1 can lead to severe metabolic issues, as it disrupts energy production in cells. So, in conclusion, complex 1 of the electron transport chain plays a key role in oxidizing NADH, transferring electrons, and pumping protons to establish a gradient. This is just the beginning of the electron transport chain, but it is foundational for the entire process of ATP generation.
Good day, everyone. I am Stefan Maurice RANCES, and today I will discuss complex 2, also known as succinate coenzyme Q reductase. So, unlike complex 1, complex 2 is smaller and simpler, containing only 4 subunits. Among these two are key enzymes involved in its function. These complex plays a crucial role in their electron transport chain by processing FADH2, which is generated in the citric acid cycle. Specifically, it facilitates the conversion of a succinate to fumarate, a step that also contributes electrons to the chain via FADH2. These electrons are then passed to coenzyme Q, helping to drive the production of ATP. The inclusion of succinate in the name highlights its direct involvement in this reaction. While it doesn't pump protons like complex 1, it is essential for maintaining the efficiency of the electron transport chain and energy production in the cell. So, continuing with complex 2, let's focus on the role of coenzyme Q or CoQ. In this complex, COQ functions similarly to its role in complex 1. It acts as the final recipient of electrons that originate from FADH2. These electrons are transferred through iron-sulfur proteins, which serves as intermediaries in this process. What's significant here is that both complexes 1 and 2 contribute to the reduction of coenzyme Q to its reduced form COQH2. This reduced form is essential because it serves as the shuttle for electrons to move to complex 3. In this electron transport chain, this collaboration between complexes 1 and 2 highlights their interconnected roles in ensuring efficient energy production. As you can see here in figure 23.13, this summarizes the electron transport chain reactions associated with complexes 1 and 2. So in figure 23.13a, the net process is shown that only starting and end product shown. But in figure 22.13b, individual reaction details is shown. So note that the general pattern that this is developing for the electron carriers, they are reduced except electrons in one step and then regenerated or like oxidized to lose electrons. So in the next step, so that they can again participate in electron transport chain reactions.
The third complex is the coenzyme Q cytochrome C reductase. Complex 3 contains 11 different subunits. Electron carriers present include several iron to sulfur proteins as well as several cytochromes. So what is a cytochrome? A cytochrome is a heme-containing protein in which reversible oxidation and reduction of an iron atom occur. A heme, a compound also present in hemoglobin and myoglobin. So this is an example of a heme that is present in hemoglobin and myoglobin. Heme-containing proteins function similarly to iron-sulfur protein. Iron alternates between +3 and +2 oxidation states. Different types of cytochromes are site A, site B, and site C. Because the iron-to-iron system involves only a one electron change, 2 cytochrome molecules are needed to move 2 electrons along the chain. So how does electron transfer in complex 3 done? First, initial substrate. Coenzyme QH2 carries electrons processed through first complex 1 from NADH and complex 2 from FADH2. Second, electron transfer process. First is the coenzyme QH2 to iron-sulfur protein to site B to another iron-sulfur protein to site C and to site C. For the third step, cytochrome C movement. Site C moves laterally in the intermembrane space. It delivers electrons to complex 4 and note that site C is the only water-soluble cytochrome. Oxidation reduction at complex 3. The initial reaction involves oxidation of coenzyme QH2 and reduction of an iron to sulfur protein. So an example of the oxidation reduction process is the figure below. So in this figure, it shows the hydrogen ions and electron transfer in complex 3. The hydrogen ions produced from the oxidation of coenzyme QH2 enter the cellular solution. All subsequent redox reactions on complex 3 involve only electrons. Electrons are conveyed further down the enzyme complex chain. So for the electron transfer steps, first is the electrons from coenzyme QH2 are transferred to complex 3. Second, they are passed through various intermediates within the complex. Third, these steps are part of the electron transfer chain, driving further reactions.
Complex 4 cytochrome C oxidase. Complex 4 contains 13 subunits, including 2 cytochromes. The electron movement flows from site C or carrying electron from complex 3 to site A to site A3. In the final step of electron transfer, the electrons from site A and hydrogen ions from cellular solution combine to oxygen to form water. 95% of the oxygen used by cells is utilized in this process. Oxygen acts as a terminal electron acceptor in the ETC. The two cytochromes present in cytochrome C oxidase, or A and A3, differ from previously encountered cytochromes in that each has a copper atom associated with it in addition to its iron center. The copper atom sites participate in the electron transfer process as do the iron atom sites, with the copper atoms going back and forth between the reduced copper 1 state and the oxidized copper 2 state. The chemistry at a glance feature is a schematic diagram summarizing the flow of electrons through the four complexes of electron transport chain. So in the picture, here's the breakdown of its components. Site C or cytochrome C. This is a mobile electron carrier that transfers electrons from complex 3 to complex 4. Iron. The iron ions in the cytochrome components or site A and site A3 plays a crucial role in the electron transfer process. Copper. There are two copper ions, copper A and copper B. The copper A accepts electrons from cytochrome C. The copper B participates in the reduction of oxygen. Electron transfer. Electrons flow site C to site A, then to iron of site A, and finally to site A3 and copper B. Oxygen reduction. At the end of the electron transfer chain in the complex form, molecular oxygen is reduced to water using protons from the mitochondrial matrix. The reaction is essential for aerobic respiration. Both hydrogen and electrons from NADH and FADH2 participate in the reactions involving enzyme complexes 1 and 2. Following the formation of COQH2, hydrogen ions no longer directly participate in enzyme complex reactions. That is, they are not passed further down the enzyme complex chain. Instead, they become part of the cellular solution where they participate in several reactions, including the process by which ATP is synthesized. So in the picture, here's the summary of the flow of electrons through the four complexes of the electron transport chain. In the complex 1, electrons from NADH are transferred through FMN and Fe clusters pumping protons in the intermembrane space. Complex 2, electrons from FADH2 enter the chain here but do not contribute to proton pumping. Ubiquinone, a mobile carrier. It collects electrons from complexes 1 and 2 and transports them to complex 3. Complex 3. Electrons are passed through cytochromes to cytochrome C and protons are pumped across the membrane. Cytochrome A mobile protein that transfers electrons from complex 3 to complex 4. Complex 4. Electrons reduce oxygen to water and more protons are pumped, enhancing the gradient. And the last is ATP synthesis. The proton gradient derives proton back into the matrix via ATP synthase produce ATP. In the conclusion, the electron transport chain or ETC, part of oxidative phosphorylation, is the final stage of cellular respiration. It occurs in the inner mitochondrial membrane and plays a critical role in energy production by transferring electrons through a series of protein complexes and electron carriers. This process creates a proton gradient across the membrane, driving the synthesis of ATP through ATP synthase. In summary, the ETC is a highly efficient process essential for converting the energy stored in NADH and FADH2 into ATP. the cell's primary energy currency. Disruptions in the ATC can lead to significant metabolic and energy imbalances, highlighting its importance in cellular function and overall metabolic health.